So you might want to take a break, take some notes in your for yourself, and now we're going to move on to lipids. So remember I said macromolecules come in 4 different flavors for us, and now we're going to move on to the second one called lipids. So lipids fall off that monomer-polymer idea, but it's still piece in many parts, if you want to say, and it's all put together still by a hydrolysis a dehydration synthesis or condensation reaction. So we're going to have the base molecule be a glycerol. Let me see if I zoomed in on that. I zoomed in on it later. Be a glycerol. And this glycerol has 3 carbons and 3 hydroxyl groups. And to each of these hydroxyl groups, we're going to save the oxygen and attach a fatty acid to it. A fatty acid is a long hydrocarbon chain. Remember we said carbons like to bond to each other, and then they can also have these hydrogens. This is very hydrophobic. The electronegativity difference between hydrogen and carbon is very little, almost the same. So they do not like water. This is not a polar bond. It's ever so slightly polar, but it's almost nonpolar. And then this has got a very different chemistry than this water-loving side over here. But if we remove this and we bury the oxygens in here, this doesn't really interact very well with water. So we're going to attach 3 fatty acids. They point out in this case this is called palmitic acid. There are many different kinds of fatty acids. There are some that are 18 carbons long. There's some that are 22, 20. We're not going to get into the names of that. You can take a biochemistry class someday and get into that. But suffice it to say they can be different lengths. We're going to have another twist in a second too. So this is, for those of you who remember some chemistry, this is called an ester linkage, a COC with a C double bond O. You don't have to worry so much about that. But we've now built a fat. A fat consists of a glycerol and 3 fatty acids. What else about fats? Well, we know fats. We see them all the time. Animal fats are called saturated fats. Butter, maybe the fat on a steak that you've seen. And here's a picture of that fat. And you notice that this fatty acid tail is nice and straight. So at room temperature, this can pack in nice and tight, and it's a nice solid. Unsaturated fats like olive oil, other plant fats, tend to have a double bond in some of their fatty acids. They're showing it in one of them here. So basically that double bond, remember we said that's a very constricted bond, that there's no rotation around it. It makes a kink and it's stuck like that. So if you have fatty acids that have kinks in here, at room temperature, when things are not moving so much, it doesn't pack in quite as much, so it stays more fluid, and it does not become a solid at room temperature. So what other kinds of lipids are there? We've talked about fats, number 1. Now we're in the second class of lipids called phospholipids. Built very much the same. They have a glycerol backbone you're going to see in a second, and then they have this hydrophilic head group called a phosphate head group. We'll see the structure of that in a second. And then they have these hydrophobic tails that are fatty acids. They can self-assemble into micelles and also bilayers, where they try to have those fatty acid tails that hate water hanging out by themselves together. And this hydrophilic head group that's going to have phosphates in it hanging out and being willing to touch water. So let's go look at that a little closer. Again, here's our glycerol molecule. You can see those 3 carbons. 1 oxygen that had an OH on it, another oxygen, and this one pops up because of that charge, doesn't like to be near these fatty acids. So again, we have our glycerol in the center. This is the phosphate head group, hydrophilic head group. It has that phosphate, remember that functional group with the oxygens and the charges. And then it's got something else. This one happens to be phosphatidylcholine, so it's got this choline molecule up here with a charged nitrogen and those other carbons. Then we do have 2 fatty acids attached to it using that condensation synthesis. And in this case, we have one that's saturated and one that is unsaturated. You know, I feel like I have to go back and talk about saturated and unsaturated for just a second. When I say saturated and unsaturated, I'm talking about hydrogens. This is a saturated fatty acid tail, meaning at each of these vertices there's a carbon, and there's a hydrogen and a hydrogen, and you see that with these bubbles. On this fatty acid is unsaturated because we had to take away a hydrogen from each of those carbons to make that double bond. and that's what makes the kink. So this has two fewer hydrogens in it, so it is unsaturated. So back to our phospholipid. Again, different phospholipids have different fatty acid tails, and since these are used in membranes, you're going to hear all about the cell membrane in 2 chapters, you can actually change the fluidity of a membrane by changing the types of fatty acids you have on its tails. Last class of lipids, so we had fats, we had phospholipids, now we have sterols. These guys don't look anything like it. They really don't fit the monomer-polymer motif at all. What they do have is this repeating 4 units of carbon ring that shows up, and then they have different functional groups attached to it. You've already seen estradiol and testosterone in an earlier screencast where I showed you that the only difference between these two, and this one actually dropped the double bond, but don't worry about that right now, is an addition of a methyl group here. So very close in shape and chemical composition, just an extra CH3 or methyl group in testosterone. Another big sterol that you hear about a lot in diet, and it's a very important part of our membranes, is cholesterol. So it has this sterol backbone here. It has methyl groups hanging off it, a hydroxyl over here. But then it has this tail, and this is a very hydrophobic tail. So this whole chunk of cholesterol is very hydrophobic. It hates water. This is little OH over here. And you're going to see how it gets embedded in membranes in the Chapter 5 screencasts. So I think we're going to stop there. Next will be proteins.